now we're going to step back and talk more about the nucleus and the material within the nucleus, which is DNA. DNA stands for deoxyribonucleic acid. It is a complicated structure that, in most cases, is loosely packed within the cells, and they wind itself around multiple different materials in order to stay accessible during normal cellular activities, but you want it still organized. When the cell goes ready to divide, it's going to then get tightly packed into the chromosome, the X-shaped chromosome that we all think of when we think of DNA. It actually is not in that X-shaped form while the cell is active, only while it is dividing. But when it is going through the division process, that DNA is going to start to tightly wrap around a material called histones. Histones are going to bundle up together in a nucleosome. Then they're going to wind together in a chromatin, and those two chromatins are going to group together to form the chromosome. Okay, honestly, this entire section from here on out should be an entirely different chapter. This is a lot of information I'm going to throw at you guys, so bear with me. Feel free to repeat this if necessary. Okay, the DNA is from a structural standpoint. DNA is made up of 4 different nitrogenous bases. Okay, nitrogenous bases are what's actually different in the DNA that makes up the sequence. We have either an adenine, a thymine, a guanine, or a cytosine. So A, T, C, or G. Those are the language of DNA. Those nitrogenous bases are then hooked together on a sugar phosphate backbone. So that's what makes the double helix stair-stepping ladder. Okay. This double helix is comprised of 2 complementary strands. So meaning if you read it one way, it's going to read opposite the other way. Okay, and they're always going to pair together on these nitrogenous bases. And it's a hydrogen bond, which we learned about in the last chapter, that's going to be connecting these nitrogenous bases. So as you can imagine, it is easy to break apart these bases, which we do when we are going to be reading the DNA or replicating the DNA, and then it's really easy to put them back together. So, DNA replication. Whenever the cell goes through cellular replication, so you take one cell and you need to divide it into 2 cells, the DNA also needs to replicate so that you have a copy of the genetic material, which is the building block of life. I consider it the encyclopedia of living cells. You need to have a complete encyclopedia for each of the daughter cells. And so when you are going to go through that process, you need to do DNA replication. During DNA replication, you are going to duplicate the entire genome of the cell. So every cell in our body contains the entire genome as a complete encyclopedia, okay? We just don't utilize the entire genome depending on what type of cell we have. The only exception to that is the egg and sperm, okay? So during DNA replication, there are some different enzymes that are going to work together in order to create this. One of those is going to be this DNA polymerase. That is going to be what's actually replicating the cell. We have what's called a helicase, and that's going to unravel the existing strand at the location of what we call the replication fork. That replication fork is the start of the unwinding, okay? The DNA polymerase is going to add the new nucleotides, either the A, T, C, or G, okay? And so when you duplicate this, you'll start with one and then you'll end up with 2 new daughter cells. Okay, a lot of information. So let's talk about the genetic code a little bit more. The genetic code holds all of the necessary information for the cell. One of the most common things that genetic material does is to build proteins. Our body is comprised of tons and tons and tons of different proteins. Proteins, as we saw on the plasma membrane, do lots of different functions. Okay, so these proteins need to come from something. So where proteins come from is instructions from the DNA. So the DNA is transcribed into RNA. RNA then will leave the nucleus and get translated into proteins by the ribosomes. Okay, let's step back a minute. So DNA is always in the nucleus. That is the blueprint of our cells. So if a cell needs to make a protein for whatever purpose it is, okay, we don't want the genetic material to leave the nucleus because that's dangerous. We want to make sure that it stays intact and safe in the nucleus, in its little bubble. So we need to have some sort of a mechanism in order to get the information out to the ribosomes. So what we do is we go through this process of RNA transcription. So DNA is transcribed into RNA. RNA is a very similar material compared to DNA, but the big difference is that instead of thymine, we have this nucleotide uracil. So instead of T's, we have U's. Okay, so RNA is transcribed from the DNA in the nucleus. That RNA is going to leave the nucleus, go to a ribosome outside in the plasma membrane or on the rough ER, and that ribosome is going to read the RNA and translate that RNA into proteins. And we have talked about a little bit the different shapes of proteins, okay? So how does transcription work? I'm emphasizing this DNA transcription, RNA translation to proteins because it's very important to remember the difference, okay? So how do we transcribe DNA to mRNA? So mRNA stands for messenger RNA. Messenger RNA is going to be that type of RNA that leaves the nucleus to find a ribosome. So whenever we have a piece of DNA that needs to be read in order to make a protein, we are going to have a protein come in that's called RNA polymerase. So very similar to DNA polymerase that we used in order to duplicate the DNA in cellular replication, RNA polymerase is going to come in and read the DNA and make messenger RNA. So it's going to transcribe the DNA into mRNA. So remember that RNA instead of T's has U's, and it's a complementary version of whatever it reads. So that's why it's the opposite of whatever is on the genetic material. So this RNA polymerase is going to run down the DNA and make the nucleotides of the RNA. Okay. Once you get the RNA, it needs to go through this process called splicing. So the genetic material has got way more information than is needed to make a specific protein. So it needs to be spliced into a readable RNA form. Okay, so this mRNA that is transcribed initially is called pre-mRNA, and it has these different regions, and not all regions are utilized. So we have exon and intron regions, okay? These intron regions are going to be removed because it is non-coding regions, meaning regions that are not important for this protein synthesis. So it's going to be removed by this material, this protein called a spliceosome. A spliceosome is going to cut out the introns, leaving just the finalized mRNA material that only has the exons that we need. There are some cases that we splice out some of the exons as well, but in this case, we'll keep it simple. Okay? So we have our finalized mRNA. That mRNA is going to leave the nucleus, and it's going to enter the cytoplasm and find a ribosome. So a ribosome is a giant protein whose job is to read the mRNA and make a protein. So remember, a protein is made up of many monomers of amino acids that are all strung together into a complex multidimensional functional unit. So this ribosome is going to go down the mRNA, and it's going to read 3 base pairs together. These 3 base pairs we call codons. The codons are all going to read for a different protein amino acids, okay? So the AUG is going to, in this case, transcribe a methionine. You guys don't need to know the specific proteins, just understand that the mRNA is going to be read by the ribosome in order to make a specific amino acid that will be strung together for a polypeptide strand. Okay, that's a lot of information. Make sure you are watching all of the videos in your book. And if you need more information on how that happens, I can either add more videos in Canvas, or YouTube is a great resource for DNA synthesis and protein synthesis. So those are the keywords that you'll be looking for, okay? So that RNA, or that DNA to protein synthesis, is called transcription and through translation. So transcription of the DNA to the mRNA is going to happen in the nucleus. Translation is going to happen in the cytoplasm by the RNA, or by the ribosomes. Okay, we're going to pause this video here because if I make them too long, they don't upload.